Hello students, welcome to Academia. Today's topic is glycogenolysis. I will discuss the process, the reaction steps and the significance of glycogenolysis. So here we start. The degradation of stored glycogen in liver and muscle constitutes glycogenolysis. The pathway for the synthesis and degradation of glycogen are not reversible. That means from glucose we got glycogen, but from glycogen directly we do not get the glucose. An independent set of enzymes present in the cytosol carry out the glycogenolysis process. Here, glycogen is degraded by breaking alpha-1,4 and alpha-1,6 glycosidic bonds. The main enzymes of glycogenolysis is number 1, glycogen phosphorylase, number 2, debranching enzyme, alpha-1,4 transferase, alpha-1,6 and alpha-1,4 glucosidase. Number 3, glucose 6 phosphatase. The reaction steps of glycogenolysis include number 1, action of glycogen phosphorylase. The alpha-1,4 glycosidic bonds are cleaved sequentially by the enzyme glycogen phosphorylase to yield glucose 1-phosphate. This process is called phosphorylasis. It continues until 4 glucose residues remain on either side of branching point. The glycogen so formed is known as limit dextrin, which cannot be further degraded by phosphorylasis. The next step of glycogenolysis is action of debranching enzyme. The branches of glycogen are cleaved by 2 enzyme activities present on a single polypeptide called debranching enzyme. Glycosyl 4:4 transferase activity removes a fragment of 3 or 4 glucose residues attached at a branch and transfers them to another chain. Here, one alpha-1,4 bond is cleaved and the same alpha-1,4 bond is made, but the places are different. The enzyme amylo-alpha-1,6 glucosidase breaks the alpha-1,6 bond at the branch with a single glucose residue and releases a free glucose molecule. The remaining molecule of glycogen is again available for the action of phosphorylase and debranching enzyme. The number 3 step is formation of glucose 6-phosphate and glucose. Through the combined action of glycogen phosphorylase and debranching enzyme, glucose 1-phosphate and free glucose in ratio of 8:1 are produced. Glucose 1-phosphate is converted to glucose 6-phosphate by the enzyme phosphoglucomutage. The fate of glucose 6-phosphate depends on the tissue. The liver, the kidney and intestine contain the enzyme glucose 6-phosphatase that cleaves glucose 6-phosphate to glucose. This enzyme is absent in the muscle and brain. So, free glucose cannot be produced from glucose 6-phosphate in these tissues. Therefore, liver is the major glycogen storage organ to provide glucose into the circulation to be utilized by various tissues. The significance of glycogenolysis is it plays an important role in the fight-or-flight response. It contributes to the regulation of glucose levels in blood. The metabolism of glycogen polymers becomes important during fasting. In myocytes like the muscle cells, glycogen degradation serves to provide an immediate source of glucose 6-phosphate for glycolysis to provide energy. In case of hepatocytes, that is in liver cells, the main purpose of the breakdown of glycogen is for the release of glucose into the bloodstream.